Is this cancer drug the real deal? Could it be the closest thing that we have to an actual cure? You've seen it on the news, you've seen it in actual research papers recently, and you've also probably seen healthcare influencers and people of a medical background talking about it on TikTok, YouTube, and Instagram. So I wanted to weigh in from a researcher's point of view, from somebody who actually did a PhD in a similar area, to weigh in and give you kind of the pros and cons and why it is something of significance, but there's a few caveats we need to be aware of. But the reason why I'm so fascinated about this in particular is actually my PhD many years ago when I did mine was on a drug discovery topic and followed very similar principles to what these incredible researchers are doing to lead to this very interesting novel anti-cancer drug. So let's get straight into it. What is AOH1996? What is this anti-cancer drug? Why is it so special? So the reason why is that it's been showing a lot of very promising results in cell culture models and in actual in vivo models about its selectivity, and this is the key part I'm going to talk about in this video. Its selectivity towards cancer cell lines and its ability to be very potent towards a cancer cell line and actually not causing much, if any, side effects towards normal cell lines. And that's the argument that's being played out in the research papers. Now, to give you a bit of context from a drug discovery point of view, many drugs can be really potent. It's quite easy to make drugs potent, but the difficulty is actually making drugs selective to what you want it to. Now, in this case, it's cancer. Now, this particular type of drug is known as a PCNA inhibitor. And all that essentially means is that it's actually inhibiting a particular antigen, proliferating cell nuclear antigen, that's actually found on all cell types, not just cancer cell types. And it's actually a DNA clamp that binds towards DNA polymerase. So in simple terms, it's something that's very important, very integral in the cell replication, cell growth phase. So which is really important for normal cell lines, but its mutant form, its kind of defective or its hyper accelerated form, it's very integral in allowing cancer cells to grow. It's a very key part found in actually nearly all cell types. So one of the recent studies actually looked at 70 different cancer cell lines, where it's looking at the ability and the effectiveness of this AOH1996. So why is it of interest? The difficulty is that in many different studies that it will show this kind of repeated result, this reliable result of being very effective at killing or destroying cancer cell lines of various different types. And that's the interesting thing. It's not just breast cancer or pancreatic cancer or whatever it is. It's actually over various different cell types. But it's showing minimal side effects and minimal toxicity towards normal cell lines. So that in essence is why this actual research or this particular drug is actually gathering a lot of interest. But let's dive into it a little bit deeper. What's so special about this particular inhibitor? So cancer therapy is very wide, very diverse. There's many different avenues, there's very different targets to look at, very, all targets, new emerging targets, different methods. You've got immunotherapy, you've got radiology, you've got different types of actual, oral medications that people use to try and combat cancer in many different regards. But why this is actually of interest is there's not many cancer therapies where it has the ability to target many different cell types, but target a very particular aspect of cancer cell growth that is found in many different cell types without causing problems to other cell types. All that means is that imagine having a target that is found on all cancer cell types, if not a good majority of them, and you have a drug that's actually got the ability to be highly selective. This is the crux of this entire video and this entire promise of this drug. It is its highly selectiveness towards this mutant integral target found in cancer cells, but it's not found in that form in normal cells. And that is this PCNA, this proliferating cell nuclear antigen. As I mentioned earlier, it's very integral in the cell replication, cell division state for DNA polymerase. So if it's an integral target in cancer cells, it's found in many cancer cells, it has very good potency in killing and stopping that actual process from occurring, and in turn cancer cells can die off because it's stuck in their replication process. But it has minimal side effects to normal cells. Now, to give you some numbers, they're looking at sometimes around 300 nanomolar in terms of its potency in killing over 70 different types of cancer cell lines via the degradation and the breakdown of that replication phase. But they've seen many minimal toxicities up to 10 micromolar. And here's the study here, so you know I'm not lying, not making it up. Here you can see on the screen right now. That's some very interesting values and numbers from a drug discovery point of view. To give you a context, this, what we call the IC50 values, something that I actually talked about in my PhD, Anything in the sub-nanomolar range, down to 300, 100, it's very, very potent. And then if to add its selectivity, makes it a game changer in the cancer therapy world, that is why this is gathering a lot of interest. And that's why a lot of people are looking at this, looking at ways to develop it. So I want to add a little bit of sense from a researcher, from a medicinal chemistry point of view, that might be of interest to many of you. This is something known as the structure-activity relationship. All it means is that, and it's fascinating to me, so I hope it comes across to you, that the original analog, so if you think about drugs, that you have a particular compound that's made and you make different variations of that compound to try and improve its activity. So AOH1996 was the initial parent compound, as we call it, and they changed and modified interactions of that molecule to better fit with the binding domains of that pcDNA and the different residues in there. You see on the screen right now, this is actually the in-depth understanding of the changes in naphthyl groups and diphenyl ether groups, and that's the in-depth understanding of how they change many different variants to produce many different analogs. AOH1996 in particular, because of its structure-activity relationship, bound better and caused better in terms of its activity. That's the notion, and that's the real-life applications of drug discovery, of analog-based drug discovery. The reason why I'm really interested in this, I talked about this, not this entire concept, but I'm very familiar with this because it's something that I talked about in my PhD, about the understanding and the benefits of analog-based drug discovery. And this is a proof of real-life kind of golden example of that in action, leading to a very interesting drug candidate that could help change the cancer. So, sounds interesting. Seems this drug actually got some potential. But what's the point? Why is this of interest? From a good point of view, we'll talk about the bad in a minute. From a good point of view, it's a promising preclinical drug candidate. Now, there are actual clinical trials where this drug is now being taken from the cell culture model to the in vivo model to now human trials to see its effectiveness in human trials. Now, in the world of drug discovery, there are many promising candidates that get to the stage, but then unfortunately fall off because of adverse side effects, because of inability to get that drug in actual physiological concentrations into the patient, or to actually off-target side effects that actually make that drug candidate a losing game, and unfortunately they die off. So a lot of drugs actually come to this stage in terms of the traditional drug discovery model, but it's actually when human trials dictate whether it's actually viable long-term. And this is the long-term game where there's many years of study, many years of understanding about how this drug could potentially improve or reveal unwanted side effects in human trials. So that's a good sign that this is now taken up into the kind of the final stages of a drug discovery window, from cell models to vivo models now to human trials. So I'll put the link down for the clinical trial that's actually underway right now. Hopefully in the next coming months, I can share an update about how this drug is actually of interest. How is it going? Is there actually some new signs? Is it more promising than we thought? Who knows? Let's find out. But here's the bad. There have been many cases, and this is not to disrespect the work that's been done in this work about producing AOH1996, but there are many principles where a drug has shown great potency, incredible selectivity, good solubility from a pharmacokinetic point of view, so it's actually not being broken down too quickly, it's not being broken down too slowly, because that's a very important part of drug discovery, making sure a drug does what it needs to do, but doesn't stay in the body too long, or doesn't actually go away too quickly. Pharmacokinetics is actually really important. There are many cases where drugs do get to this stage, and it differs or causes problems once it gets to human trials. Because no matter how sensitive or how accurate or how complex cellular models, 3D models, molecular docking, in vivo, advanced cellular models, it doesn't replicate to the best degree what humans go through in terms of absorption, how it gets into the cells. All of those other things is only replicable if you do it in human trials. So although this has gathered a lot of interest, and rightly so, like many other drugs, we have to be cautious and take it with a pinch of salt, because things can drastically change and differ in human trials, because there are many other side effects that could occur because it's now going through the body. It's not being just handed directly into the cancer cell. If it's taken orally, or even from an IV point of view, it's now going to encounter other aspects that you probably might not find in an animal model, you might not find in cellular model. So there's now more unknowns this drug needs to go through in order to be vetted and be potentially viable as a licensed drug therapy. So I hope you find this video useful. I know it's a different type to what I normally do, but I do enjoy, being a researcher, putting my researcher hat on and delving into the science about what's currently in today's news helping it to distill it in a nice format for you to enjoy. But now, when I was making this video, I was quite happy but also quite sad, because we might see that this drug is incredible, there's a lot of research and a lot of exciting research happening, but I know from behind the scenes what some of these academics, some of these researchers who are doing the grunt work for day in day out might be going through. Now, if you want to understand what I mean by that, is I made a video here about how academia is broken in my opinion, and how despite a lot of research coming out, the culture needs to change. There needs to be more respect, there needs to be more appreciation for the people who are doing this incredible work. If you understand what I mean in a bit more detail, check out that video here, and you'll understand what I mean by things need to change. I hope you find this video useful. Thank you for watching. Do let me know if you've got any thoughts in the comment section down below. Consider subscribing if you want to. I'll see you in the next one.